We're going to talk about understanding penicillin and bacterial resistance. We're going to explore how penicillin kills bacteria and how bacteria have evolved mechanisms to resist its effects. Antibiotics are essential in modern medicine for treating bacterial infections. They have significantly reduced morbidity and mortality rates associated with bacterial illnesses, saving countless lives. Antibiotics enable healthcare providers to effectively manage a wide range of infections, from common ailments to life-threatening conditions. They are crucial in medical procedures such as surgeries and organ transplants, where the risk of infection is high. Additionally, antibiotics have contributed to controlling the spread of infectious diseases and improving public health. However, the rise of antibiotic resistance poses a serious threat to our ability to treat infections effectively. It is imperative to use antibiotics responsibly and develop new strategies to combat antibiotic resistance to ensure their continued efficacy in saving lives. Okay, we're on how penicillin kills bacteria. Penicillin is one of the most widely used antibiotics. It is classified as a beta-lactam antibiotic. It exerts its bactericidal effects through a fascinating mechanism of action. Let's delve into how penicillin targets and eliminates bacterial cells. Firstly, it's important to understand that bacteria possesses a rigid cell wall that provides structural integrity and protection. This cell wall is primarily composed of a molecule called peptidoglycan. As bacteria grow and divide, they continuously remodel their cell wall, a process crucial for their survival. This remodeling involves enzyme transpeptidase, which cross-links peptidoglycan chains to strengthen the cell wall. Here's where penicillin comes into play. Penicillin works by inhibiting the activity of transpeptidase. By binding to the active site of transpeptidase, penicillin prevents it from cross-linking peptidoglycan chains properly. The result, the bacterial cell wall becomes weakened and structurally compromised. Without a robust cell wall to maintain its shape and integrity, the bacterium is unable to withstand osmotic pressure and undergoes lysis. In essence, penicillin disrupts bacterial cell wall synthesis, ultimately leading to destruction of the bacterial cell. This mechanism of action highlights the precision with which antibiotics like penicillin target specific bacterial processes, making them indispensable tools in combating bacterial infection. All right, we are going to delve into the critical impact of penicillin on cell wall synthesis. The bacterial cell wall, primarily composed of peptidoglycan, provides structural integrity and protection, with penicillin inhibiting the action of transpeptidase, a key enzyme involved in cross-linking peptidoglycan chains. The bacterial cell wall becomes weakened and compromised. This disruption in cell wall synthesis renders the bacterium vulnerable to osmotic pressure, like we just discussed, leading to cell lysis and ultimately bacterial cell death. Understanding this process underscores the precision with which antibiotics like penicillin target specific bacterial structure and processes, highlighting their effectiveness in combating bacterial infections. So this is the chimera image. Penicillin, a beta-lactam antibiotic, exerts its antimicrobial effects by inhibiting the enzyme transpeptidase. This enzyme is crucial for, like I said, bacterial cell wall synthesis, as it catalyzes the cross-linking of peptidoglycan chains, providing structural integrity to the cell wall. Penicillin contains a beta-lactam ring that structurally resembles the D-anylyl D-aniline portion of the peptidoglycan precursor. When penicillin binds to transpeptidase, it occupies the enzyme's active site, preventing it from interacting with the peptidoglycan precursor. As a result, transpeptidase is unable to catalyze the cross-linking reaction, leading to the formation of structurally weakened cell walls. This disruption, like we just said for the third time, ultimately causes bacterial cell lysis and death. The bound structure of penicillin exemplifies the specificity of its action, targeting a key enzyme involved in bacterial cell wall synthesis and highlighting its importance as a therapeutic agent in combating bacterial infections. We're going to explore the broader context of antibiotic resistance. Antibiotic resistance poses a significant challenge to global health, threatening our ability to effectively treat bacterial infections. Bacteria have evolved various mechanisms to resist the effects of antibiotics. antibiotics, including changes in cell permeability. These mechanisms may involve modifying cell wall structures, developing penicillin binding proteins or PBPs with reduced affinity, producing beta lactamase enzymes to degrade penicillin, or upregulating efflux pumps to expel antibiotics from the cell. By understanding these resistance mechanisms, we can better appreciate the urgency of combating antibiotic resistance through responsible antibiotic use, infection prevention strategies, and the development of novel antibiotics. We're going to examine the diverse mechanisms bacteria employ to resist the effects of antibiotics. Antibiotic resistance is a complex phenomenon driven by genetic changes in bacterial populations. One common mechanism involves alterations in cell permeability, which hinder the entry of antibiotics into bacterial cells. Bacteria may modify their cell wall structure but decrease the expression of porins or upregulate efflux pumps to expel antibiotics from the cells, like we just said. Additionally, bacteria can develop enzymes such as beta lactamases, which degrade antibiotics like penicillin. These resistance mechanisms pose significant challenges in the treatment of bacterial infections and underscore the urgent need for novel strategies to combat antibiotic resistance and preserve the efficacy of existing antibiotics. We conclude our discussion by emphasizing the critical importance of understanding antibiotic resistance and its implications for healthcare. Antibiotic resistance poses a significant threat to public health, limiting our ability to effectively treat bacterial infections. By comprehending the diverse mechanisms bacteria employ to resist antibiotics, we can develop strategies to combat resistance and preserve the effectiveness of existing antibiotics. This requires a multi-faced approach, including promoting responsible antibiotic use, implementing infection prevention measures, and investing in the development of new antibiotic and alternative therapies. Addressing antibiotic resistance is a collective responsibility that requires collaboration among healthcare professionals, policymakers, researchers, and the public. Together, we can work towards preserving the efficacy of antibiotics and safeguarding global health. Thank you so much.